Emanuela, and go ahead, please. Thank you.
Hi, thank you very much for the introduction, and thank you for having me participate to this wonderful meeting. I'm very sorry not to be able to be there in person. So what I want to talk about is our attempt to try to understand the complex rheology of particulate gel networks, and in particular what we have managed to or try to understand in connection to their complex rheological response. So before I dive into that, let me just give you a quick overview of my group so that you can also maybe see what are the specific interest and the perspective that we have in trying to understand these issues. So in general, we are a group doing a lot of computational research. What we are mainly interested in is complex materials in which we need to understand the development of microstructural elements and larger scale structural elements, which then have an impact in the time scale and length scale that are embedded in these materials, and that enters the way these materials age, the way these materials are processed, and various phenomena that are characteristic of these materials, from yielding, memory, thixotropy, and so on. And one of the reasons that we are interested in understanding this is because, of course, there is a number of very interesting physics questions, and you will hear about them in a minute, but also we are interested how this type of understanding, of fundamental understanding, in the end is crucial to design sustainability both of materials that we want to use and the processes that we use to make materials, to process materials, or to subject materials to when they are in the environment. Here you have a little overview of different type of questions and systems that we study, and feel free to contact me or to ask me a question in the end of the talk if you want to know more about these different stories. Okay, so the work that I'm going to talk about, in fact, it's a team effort, and it's been a collaboration with a group of fantastic people, and particularly with Thibaut Divou and Mehdi Bouzid and the group of Margaret McKinley at MIT. In particular, I want to thank Minas Pibantawa, that was a grad student with me throughout all these projects, and now is a postdoc at UT Austin, and Bhavan Keshavars, that started in this project as a grad student also with Garrett, and is actually starting soon as assistant professors at Duke University. Okay, so they did really all the work, and so they should be praised for it. Okay, so basically what we are interested in is the formation of soft particulate gels, which are gels that occur and form and are relevant to all sorts of phenomena and all sorts of materials, and because they occur when you have particles, small particles, nanoparticles, micron-sized particles that are in a solvent, and they interact mainly through attractive interactions mediated by the solvent. And through these interactions, they form spontaneously self-assemble into these complex gel networks. So you have a material that in the end can be very dilute. So this is one of the interests in this type of materials, is that in the end you can have a solid material or with a tiny amount actually of solid, since this gel can form at particle volume fraction that are very, very small. So you end up with these matrices, like you see examples here, which are very porous, disordered, complex, in certain cases really hierarchically assembled. And they, as I was saying, they are relevant to a broad range of materials, a broad range of technologies. And one thing that is very interesting, intriguing, and annoying at the same time is the fact that differently from gels that, for example, we know more, like biopolymer gels or polymer gels, where there is a more clear structure that we can then try to relate to the rheological properties. These structures in this type of materials, through this spontaneous self-assembly that can happen in very different conditions, they can be very messy. It's very difficult to find common patterns, to see if there is self-similarities, and it's actually very difficult to predict from the structure the type of rheological properties. and it seems even that there is no actually organizing principles in the end, that for each gel, I have to think about it in a very, independently, as if it was a new material. And the fact that the material is gelling and is reaching mechanical stability and rigidity doesn't help me to understand something more general about the rheology. So the question that we've asked ourselves is this really like that, or there is more that we can try to understand? Okay, so basically what we have done is to develop, to use a model that we have, a microscopic model that we have developed over the years to be able to study this type of materials in numerical simulations. So I'm not going to tell you much of the model in this talk, but I'm happy to provide more information, and I have some backup slides in case people want to know more questions, have more questions. So, but the idea of the model is that I have particles, which are the units that form the gel. They can be small particles, they can be aggregates of particles, as sometimes it happens in certain systems. And then these particles, they stick together due to these sticky interactions. So we can use them in a computational model that basically can handle a large amount of particles, and it can have the gelation happen through the aggregation of these particles or of these structural units. When the particles stick together, there is, of course, the bond, if you want, that it's a physical bond that it's formed through these sticky interactions. And what we also have in the model is that the interactions are written in a way that when particles start to stick together, there is also some resistance or some price to pay for the particles to roll over each other. And this is to mimic the fact that the contacts between these particles when the gel forms can be actually more complex, and due to the heterogeneities on the surface of the particles, to the maybe different shapes of these aggregates, and to adhesion that can happen really at the surface of the particles. So through this model, now I can perform numerical simulation where I watch the particle aggregates, and I can change the interactions between the particles at different speed, so that I have I can use this to mimic different gelation rates, right, as in fact it happens in the experiments. So here what I've shown here are 3 snapshots from these simulations, where I'm showing you only the network of bonds between the particles, just for clarity. And the reason why I'm using this is because I want actually to highlight the fact that through this disordered mixture of equilibrium and non-equilibrium processes aggregation, I end up with something that it's really very messy, where there is you can see that there is a high degree of heterogeneity in the way this network is actually spatially organized. And the colors that I'm using here, I will come back to that in a couple of slides, but it's a measure of this degree of heterogeneity in the topology of this local topology of this network. And so what I'm doing here is that I am in the simulations that I'm producing these type of structures through a process that mimics as close as possible what would happen in a typical experimental system. And then I can perform basically numerical simulation in which I mimic rheological linear viscoelastic tests. So when I'm wiggling the material back and forth to obtain the whole viscoelastic spectrum. And so what you see plotted beneath the snapshots are, our viscoelastic spectra, so G prime and G double prime, the storage component and the loss component of the modulus of the material as a function of the frequency when we vary these frequencies over a broad range in the simulations. And so then you can see that actually this parameter phi that I have here is actually the volume fraction. So as I was mentioning before, volume fraction, so the total volume of the box that is actually occupied by the particles, this can be very small, as it is in fact in the experiment. And by varying the volume fraction of a relatively small amount, I can see that in fact the modulus can change by several orders of magnitude. And the heterogeneity in the network structure changes also a lot. But I can fix the volume fraction, which I'm doing here between the center figure and the right hand figure. And in fact, I can I can obtain, I can also vary the degree of heterogeneity in the network enormously, and therefore also the rheological response by changing the rate at which gelation happens, which is this other parameters that I'm indicating here. So with this model, I can in fact basically obtain a huge variety of different structures, kind of similar or mimicking what in fact I observe in the experiment. And we've studied the aggregation, the gelation in this model and the properties of these gels, both the microscopic dynamics, the structure, and compare them, and the mechanics and the rheology in various regimes, and compare them with experiments in a series of papers that are listed here. And the other thing that I wanted to add is that the way we performed the simulation was using basically extending to the simulation an approach that was developed by our collaborators for experiment, where instead of running this rheology simulation by fixing one frequency and doing the oscillation at one frequency and then going to the next frequency and so on, using chirped signal. And so if you do that, that's quite interesting, because you can then basically compute the whole viscoelastic spectrum in one simulation. And this now multiplies our capability to compute interesting properties of these materials. Okay, so what we did is, in fact, what probably you were thinking, so generate a lot of these gels at different volume fractions and with different gelation rates, and then compute the spectra. So here what I've plotted as a function of the frequency is tan delta, which is also called the loss tangent, and it's the ratio between G double prime and G prime at any given frequency. So it's the ratio between the capacity of the system to dissipate or versus storage energy. And so you can see that very much to what I was showing you before, G prime and G double prime, also tan delta displays these main features, that these spectra of response are very broad. So there is a huge range, quite a large range of frequencies, several decades, that I need to consider to understand, to capture the full rheological response. And also changing the volume fraction, even if in a small range, and changing the gelation rate in a relatively small range that we have been able to do in the simulations, you get quite a variation in these viscoelastic modulus, and we know that this comes from the structure. Okay, this seems to be all over the place, but actually you start to notice, or maybe people that have stared at this type of signal for a long time, many a lot of experience in doing that, they start to notice that this spectra, they sort of look similar, because there is this sort of more power law behavior, and then there is approach towards something that looks like a plateau. So there comes this idea that maybe I can, in fact, rescale the frequencies, so it can rescale the x-axis to get all the spectra to superimpose onto a unique master curve, and then I can use, in fact, the same scaling, the same rescaling that I use on the x-axis to then use that for my complete viscoelastic spectrum, and then rescale the y-axis of G prime and G double prime to actually obtain a whole master curve for the whole viscoelastic spectrum. So this seems to be very working extremely well. And what is interesting in doing this type of constructing this type of master curve is that now I can try to understand, okay, what is sort of the organization principle that allows me to do that? So what is the way, the mechanics of the spectrum that of this unique form of the spectrum that the gel seems to belong to? What does it mean? So the first thing that I can do is to think, okay, there is some, I can represent this, the rheology of this complex network in terms of some viscoelastic elements. And then I realized that if I was just using, and from the shape of the spectrum, I can think, okay, this is some Kelvin-Voigt type of behavior from the shape here, the lowest frequencies. And if I look at, using a Kelvin-Voigt type of element to recapitulate the spectrum, it's very clear from here, from the tan delta, that I can only capture the very low frequency end of this. But there is a deviation from this Kelvin-Voigt, which is actually very interesting. So I start, which says there are more time scale and length scale, therefore, in these systems. I can start to include more element. And then I realized that, in fact, the only way to do this, to capture the whole disk profile, in fact, is consider a huge amount of these elements, but also I really have to organize them hierarchically. So the shape of the spectrum and, in fact, the shape of the master curve that derive for the whole spectrum that derives directly from this, in fact, tells me that there is clearly a signature of self-similarity and the hierarchical organization in the microstructure, in the rheological response of the systems, in spite of the fact that the structures seem so messy and not obviously self-similar. And so this type of rheological model, which consists of these viscoelastic elements, which are organized in a hierarchical way, in a ladder way, it's been used, in fact, typically to describe the behavior of fractal structures. In cases in which I know that there is a fractal structure, for example, polymer gels typically close to the percolation transition, because I know that the chemical architecture has to have some fractal properties, and that is what will explain the self-similarity of the rheological response. What is interesting is that, in fact, in here, we have all the structures. We actually can try to look if there is something self-similar in these structures. And if we do just box counting in our gel, we don't see any self-similarity. And I showed you the images, the snapshots of our gels. So, and it's also similar to the snapshots that we see sometimes from confocal microscopy that can be used to image the structure of the systems. It's very hard to see any self-similarity or to obtain a clear signal of self-similarity from those structures themselves, even if you have access to images of the structure. Okay, so the question is what's going on, and there is more that the rheology is actually capturing about the way the material is organized that goes beyond what we can in fact see. Here, what I've done is that I have, I can switch from this description of this discrete element to more compact representation of this type of rheological response, going to a rheological fractional model where I use basically fractional derivatives to recapitulate this hierarchy of structure that I have in the discrete ladder model. Okay. So the idea was that there may be some structural characteristics in the gels, which is related to this self-similarity that I do see in the rheological response, but it's not visible simply just accessing the structure of the gel. And the way we've thought about this is that, in fact, the degree of heterogeneity in the network topology plays a very important role in connecting to the rheology. And we've come to this idea because we've studied a lot for a long time the microscopic dynamics in the system and the rheological behavior under very different conditions. So we know that the degree of heterogeneity, the fact that you have regions that are very densely connected interspersed with regions that are very poorly connected, in fact, it's a fundamental aspect of how the type of microscopic dynamical modes that you can have in these systems, but also of the type of rheological response. So what we do is that we measure the distances. So we take two branching points in the network that you see here, and we march along the network and we measure the shortest distance along the network that connects two branching points, and we obtain a whole distribution of those because, as you've seen, these networks are so complex and so messy, and then from this distribution we actually take the variance. So we see that this distribution, in fact, becomes broader and broader because as you go to lower densities, these networks become more and more heterogeneous. Or if you go to, if you vary your gelation rate, they can become more and more heterogeneous. And so this distribution and the width of this distribution actually characterize that. From the width of this distribution, we can in fact extract a length scale. We can turn that into a length scale. And so if we look, for example, at the microscopic dynamics. So here I'm looking at the fluctuation, the displacement, the fluctuations of the microscopic displacement of the particle and the distribution of these fluctuations, again, obtained for a broad range of gels. And they change, they vary a lot and they vary all over the place. But if I use this length scale, I can actually rescale all this distribution into one, telling me that in fact the difference that is expressed that is quantified by this length scale in fact captures the huge differences in the microscopic motion in this type of system. So the idea is that this degree of heterogeneity in the network topology that we measure through this length scale in fact contain basically it's key to what we are trying to understand because this length scale in fact contains information about what is the local environment, but also what are the larger scale heterogeneities that I have in this type of network if I change the gelation condition and the amount of particles. Okay, so the idea now is that this length scale actually can give me access to the type of structural element that are organized in a self-similar fashion and that maybe are what the rheological signal, the rheological response is providing insight into. So how do I actually convince myself that there are this sort of fractal elements that are hierarchically organized in my system, even if I cannot directly see them, right? So what I do, for example, here is that I can measure this length scale. If you look first in the inset of this figure, if I measure this length scale, for all these different gels, varying the volume fraction or varying the gelation rate, then okay, I can see that it varies in different ways, depending, along all these gels. But in fact, if the idea is that all these gels maybe can be recasted or re-understood in terms of some characteristic properties that they have. And this characteristic property is, we think that it is the distance from their rigidity threshold. So in this type of model, I can use a proxy to understand what is the rigidity threshold. And this proxy is basically the amount of branching points, because in the limit, as my network becomes more and more dilute, more and more floppy, more and more poorly connected, then the amount of branching point basically goes to zero. And I can obtain different amount of branching points either with a given volume fraction of particles, but also for the same volume fraction of particles, changing the gelation rate. And so the idea is that no matter what I do, in fact, by the amount of branching point will tell me how far these gels are from a gelation threshold. So I'm using this phi-BR as the distance, a way to measure the distance from their rigidity percolation. And if I do that, and if I plot my the size, the length scale that I've extracted from my microscopic dynamics as a function of the amount of branching point, then I do see that all the data for all the gels, they sort of scale onto a unique power law behavior, which looks like some kind of power law behavior. So now I start to have more confidence that maybe this length scale, in fact, tells me it corresponds or captures the scaling of a correlation length, that in fact it's increasing close to the rigidity transition. Right. So basically, this phi-BR is telling me about the distance from a rigidity transition, and the length scale that I've extracted from the heterogeneity in the topology tells me about the rigidity transition, because it's the correlation length associated to that type of, if you want, rigidity percolation transition. The exponent that we get from here seems actually relatively close to what I would obtain for a random percolation in 3 dimensions. So that's more or less the picture that I start to build up. And I can test it more, because now if I think that, okay, all my gels are rigid, all the ones that I'm considering, G prime was always larger than G double prime at the lowest frequencies that I've sampled. So basically I can think that now my blobs that I do have blobs that are fractal, and they are size Xi, and these blobs are embedded in the structure of the system, and they come from the fact that when the system first became rigid, it became rigid through this type of rigidity percolation, which looks a lot like a random percolation in our case. And so if now I have a gel that is made of these fractal blobs, right? Then the scaling, and if I do a box counting, then the scaling that I would expect, it's not simply that the mass in a box of size L, which is, let's say, my simulation box, should scale like L to the df, because this would be only true if my size of my system is smaller than the correlation length. But the correlation length here, the size of my blocks, my blob is always smaller actually than the size of the box, because I am past this rigidity transition. So I should see this type of scaling. And so in the inset here, I have the plot that I can obtain from the box counting if I do basically if I just look for the first type of scaling, right? So the exponent that I get, it's 3. This is what I was saying before. I don't see any self-similarity. But if I use instead this type of scaling, then I obtain basically my data to collapse, and I obtain that the fractal dimension of these blobs would be actually around 2.5, which is, again, seems quite similar to what I would obtain for random percolation. So now I start to believe that maybe there are blobs of a certain size xc or proportional to this size xc, which have which are fractal, and it's the hierarchical organization of these blobs that actually controls the rheological spectrum. And this is what I see when I perform my rheological experiments, even if once that I've gone beyond the rigidity threshold, I cannot actually see that anymore. So, okay, now let me see if I can test this further, right? So if I think that my blobs are actually fractal and they fill the space, it means that the dependence of xi on the volume fraction of the particle should be of this type, right? This is just the usual argument. I have a certain amount of particles which correspond to a certain volume fraction, and I make blobs of fractal dimension df in the dimensional space, and so that's the relationship between the size of the blobs and the amount of particles that I have. On the other hand, if this is true, right, and if xi is indeed the correlation length of my system as I approach the rigidity threshold as phi br goes to 0, this implies that there should be a relationship between the amount of branching point and the amount of particle in my system that is described from this type of scaling. And so indeed, I can, if I use the nu that I have obtained from here, and I knew the df that I have estimated on the basis of my box counting scaling, then I obtain this type of prediction, and these are the data that indeed I measure from my simulation. So this again strengthened my conviction that this picture is really corresponds to the physics of something that is happening in my system. The other line that I have here is are the type of scaling that I would obtain if I was considering some specific aggregation kinetics like the LCA or RLCA, which are indeed observed in some colloidal systems, and I will come back to that later. So this the type of scaling that we obtain, and in this model we rather have some kind of just random aggregation, and so then this is what we see. Okay, so the question is, why are these fractal blobs that are embedded in my structure, why are they relevant to the rheological response of the system? And the idea is the following, that in fact, if I go back to this sort of blob links and nodes model that people had developed for a fractal percolating cluster close to a percolation transition, right? What was very much important in this picture, what I think one very important insight is that not all the bonds are basically created equal, right? There is a hierarchy of bonds, and in fact, there are certain bonds especially which are what one would call the singly connected bonds, which are bonds that if I remove them, then I interrupt the connectivity, because this was the ideas built for connectivity percolation. But I can think in the same way in terms of rigidity in this case. And so what happens to these singly connected bonds in this blobs, links, and nodes picture is that they are in fact hierarchically distributed over my system, and which means that whatever length scale I pick, there will be always singly connected bonds that are there. And in particularly close to the percolation threshold, that's what happens, that any length scale I pick, there will be the probability of finding one of these singly connected bonds is finite. And that's why, in fact, the amount of singly connected bonds tells me directly about the distance from the percolation threshold, or in the rigidity threshold in this case. So basically, the reason why this fractal blobs play a role, because they tell us about the presence of these singly connected bonds, and it's their distribution that it's in fact hierarchical in the system, because it's directly related to the distance as you go close to the rigidity threshold. So now basically I can go back and think about the elasticity, for example, of a network that is so organized. So if these blobs that are fractal and are composed of this hierarchy of singly connected bonds, if these blobs are in fact growing in size as I approach the percolation threshold, and therefore this is the signal of, and this corresponds to the increasing of the presence of the singly connected bonds across the system. And basically, if these are the mechanical elements that are responsible for the rheology, then I can think in terms of one of these. So at the rigidity threshold, we have basically one blob and one link that spans everything. And I can try to understand the typical modes of these links, which are related to the distribution of bending stiffnesses that I have as I go, as I consider one bond and I start to go away from this bond in this link, because I will have different degrees of bending stiffnesses depending again of how these singly connected bonds are distributed. And so if I look at the hierarchy of these modes, in fact, I realize that I completely recover the hierarchical organization of the ladder, of the rheological ladder model that I had used to fit my master curve. So basically, I've closed the loop, and going from what I see first in the rheology, I can go back to the microstructure. I can make the hypothesis of these blobs. I can corroborate this hypothesis looking at the scaling properties that I should be able to see once that I know that are there. And then from the hierarchical organization of this mechanical element, I can actually go back to the ladder model that I was using to fit my structure, my rheological response. So why this is interesting? Well, if this is true, now I can think of my network, my gel network has in fact, in terms of these fractal blobs. And now we can see, okay, these are fractal blobs of springs, and these springs percolate at the rigidity transition. So I can use, in fact, theories developed for elastic percolating networks to estimate how the modulus of this of the gel, once that the springs, the connection of all these springs percolate, and therefore how this should scale as a function of the distance from the rigidity threshold, as a function of the distance from the rigidity threshold. And here we use a model the prediction of a model that was developed for springs with bending stiffnesses, because this is what we have here. And so basically, the models should scale with this correlation length with this type of exponent, where f was estimated in this way from the critical exponent of the correlation length. And for the yes.
Sorry to interrupt. This has been great. We are a bit behind the schedule. Can I ask
Okay.
you to wrap up like in
Yeah. This
2
is
minutes?
basically my last slide.
Okay, thank you.
Okay.
Okay.
Sorry if I went too long.
That's
I
all right.
couldn't see
That's all
what
right.
you were
Yeah.
making sound. Yes. So basically, this is the prediction that we make, and this is the prediction that we have for the exponent. And now I can replot my modulus and the modulus that I obtain either measuring the modulus or by shifting the vertical axis of my viscoelastic spectra, and the time scale that I obtain by shifting the horizontal axis of my viscoelastic spectrum, and I can plot them as a function of the distance from the viscoelastic, from the rigidity threshold, either in terms of the amount of branching point or in terms of the correlation length. And I see that basically this quantity follow exactly the prediction of this. So what I was introducing is basically the scaling of the modulus that I obtain here, which is predicted by this model. But what we find, which is a new result, is that this time scale that I use to rescale the x-axis in my viscoelastic spectra should scale in the same way. And there are some interesting ideas of why this happens. It's never been tested in experiments, so it's also an exciting prediction that comes out of looking at things in this way. The other thing is that now I realize that, in fact, when I measure the modulus in the experiment, I don't have access to this distance from the rigidity threshold, right, in the experiment. And so I only can measure the length or the whatever property, like the modulus of the system, as a function of the volume fraction. But what this picture is telling me that, in fact, the scaling that I measure as a function of the volume fraction will be different or will be related in different ways to the distance of the system from the rigidity threshold, depending on what is the system, the distance of the system from the rigidity threshold. So depending on what is the relationship between the correlation length and the amount of volume fraction, or the amount of branching point and the amount of volume fraction. And so depending on different aggregation processes, this relationship here can be different, which then can change the exponent that I measure. And so this type of scaling actually allows us to recapitulate the fact that in experiments, I can have a very different type of exponent in this type of dependence that can be measured in experiments. And so what we are saying is that this different type of exponent, which have so far always been interpreted as a lack of universality, can be reconducted to in fact, the presence of universality and self-similarity, if I consider that actually, depending on aggregation processes, I can have always the same f exponent that identifies the scaling as a function of the distance from the rigidity threshold, but they can appear as a different exponent because I have different aggregation processes. And so this is something that, in fact, has a very interesting connection with the experiment. And as I promised, I'm done, and this is just my summary. And I let you basically read that while I conclude, and maybe I ask for a question if there is the time.
Okay, thank you. Let's thank Emanuela. We have time just for one question, maybe, but
So thank you, Emanuela. Actually, when I think about a gel, it's not surprising that you have a kind of a fractal structure, right? I would not expect to have three in this gel. So actually, I'm surprised that you have such a big the fractal dimension. Wouldn't you expect to have a different fractal dimension if you decrease, let's say, the fraction or the, I don't know, I mean, depending on the some type of control parameter, I would expect to have the different fractal dimension, right? Do you observe that or not?
Yeah, so this is a little bit the point of the slide where I was showing the different exponents that you would obtain, because in fact, so what happens in this system, I believe, is that you can have a fractal aggregation from the beginning, which will be more, let's say, similar towards DLA, right? So there I would have a smaller fractal dimension, right? And then these structures that form, for example, through this type of aggregation could be the basis for these fractal blobs that we talk about that then enter the rigidity percolation. And so the fact that you have this smaller fractal dimension then would show up in a different exponent of how the modulus increases with the volume fraction of the gel. Exactly because with the different type of fractal aggregation, this is one mechanism where I can, in fact, so different aggregation kinetics, I should be able to play with that as you are suggesting. The other question is whether when I do this, actually, what does this change in terms of the rigidity percolation? And I think this is more mysterious because I don't think there is a clear understanding of how the what is the direct role of fractal structures in the rigidity percolation. So this is basically a first attempt to build that picture. I hope I answered a little bit to this question.
Let's thank Emanuela again. I have bazillion questions for you, but we'll
I know,
leave for later. Okay,
Yes.
thank you.
Thank you. Thank you, everybody.